What is qualitative analysis? Answer is qualitative analysis involves identification of compounds based on their chemical and physical properties. What is quantitative analysis? Answer is quantitative analysis involves estimation or determination of the concentration or amount of chemical compounds. What is equipment? Answer is equipment consists of one or more instruments to perform different kind of activity. What is instrument? Answer is instrument is a part of equipment. The instrument is measures the data. What is calibration? Answer is the particular instrument or device produces results within the specification limits by comparison with those produced by traceable standard over an appropriate range of measurements. This is the definition of calibration. What is friability test? Answer is friability is used to durability of the tablets during transmit, or friability measures the difference between weight of sample before and after taking in order to check the physical strength of the uncoated tablets. What is disintegration test? Answer is disintegration test measures the ability of the tablets break down into smaller particles or granules. What is hardness test? Answer is hardness test measures the breaking point of the tablet is based on its shape. Define normality. Answer is the number of grams equivalent of substance dissolved in liter of the solution. It is denoted by capital N. Define molarity. Answer is number of gram moles of solute for the liter of the solution is called molarity. It is denoted by capital M. What is molality? Answer is number of moles of substance dissolved in kg of solvents. It is denoted by small m. How many types of dissolution apparatus? Answer is as per USP we are having 7 different types of the dissolution apparatus. First one is basket type. Second one is paddle type. Third one is reciprocating cylinder. Fourth one is flow through cell. Fifth one is paddle over disc. Sixth one is rotating cylinder. Seventh one is reciprocating disc. What is dissolution acceptance criteria? Answer is for stage 1 Q plus 5% for all units. For stage 2 Q minus 15%. For stage 3 Q minus 25%. What is the USP general chapter number of the dissolution? Answer is dissolution USP general chapter number is 701. What is data integrity? Answer is when the data is in complete consistency, reliability, trustworthy throughout the life cycle is called data integrity. Data integrity clearly indicates the quality. Quality means doing right when nobody is looking. What is Alcoa and Alcoa Plus? Answer is A stands for attributable, L stands for legible, C stands for contemporaneous, O stands for original, A stands for accurate. Complete, consistent, enduring and available. These 4 points are coming under the Alcoa Plus. What is the principle of HPLC? It is a technique used for the separating of the mixture of the compounds into individual components based on adsorption, partition, ion exchange and size exclusion. Stationary phase and mobile phase are used in it. HPLC is used for the identification, purification and quantification of the compounds. What are all the types of HPLC detectors? Answer is UV visible detector, refractive index or deflection index detector, fluorescence detector, electrochemical detector, mass detector and photodiode array detector. What is the abbreviation of US FDA and MHRA? US FDA stands for United States Food and Drug Administration. MHRA stands for Medicines and Healthcare Products Regulatory Agencies. What are all the US FDA? Form 482, 483 and 484. Answer is 482 stands for Notice of the Inspection Form. 483 stands for Inspectional Observation Form. 484 stands for Receipt of the Sample Form. What are all the long-term testing stability zones? Answer is Zone 1 temperature zone, Zone 2 is subtropical zone, Zone 3 is hot dry zone, Zone 4A is hot tropical zone, Zone 4B is ASEAN testing condition zone. What is the cations and anions? Tell some examples. Answer is the ions with a positive charge is called as cations. For example, sodium and potassium. The ions with a negative charge is called as anions. Hydroxide, bromide. These are all the example for the anions. Explain Beers-Lambert law. For a given material, sample path length and concentration of the sample are directly proportional to the absorbance of the light. Formula is A equal to ELC. Which gases are using in the gas chromatography? Answer is nitrogen, hydrogen and helium. These three gases are using in the gas chromatography. Why nitrogen and helium are using as a mobile phase in gas chromatography? Answer is nitrogen and helium are low molecular weight and chemically inactive. So these two are using mobile phase in a gas chromatography. What is 21 CFR? Answer is Food and Drug Code of Federal Regulations. What are all HPLC system suitability parameters? Answer is tailing factor, theoretical plates, signal to noise ratio, relative standard deviation, resolution, retention time, peak to noise ratio, correlation coefficient and correction factor. What are HPLC parameters? Answer is efficiency factor, retention factor and separation factor. What is the principle of IR spectroscopy? Answer is when energy in the form of IR is applied, it causes vibrations. Applied IR frequency equal to natural frequency of vibrations. The absorption of IR takes place. Finally, the peak is observed. What are uses of IR spectroscopy in the pharmaceutical industry? Answer is identification of functional groups, structural elucidation, identification of substances and detection of impurities. What is UV range and visible range? Answer is UV range is 200 to 400 nanometer, visible range is 400 to 800 nanometer. Below 200 nanometer means we are identifying by X-ray. What is spectroscopy? Answer is spectroscopy is a study of interaction between matter and electromagnetic radiation. What is spectrophotometry? It is a branch of spectroscopy that deals with the measurement of radiant energy transmitted by the body function of the wavelength. What is chromatography? Answer is chromatography is a separation technique used to separate individual components from the mixture of the solution. What are strong acids and weak acids? Answer is sulfuric acid, hydrochloric acid, perchloric acid are strong acids. Acetic acid is a weak acid. What are strong bases and weak bases? Answer is sodium hydroxide and potassium hydroxide are strong bases. Ammonium hydroxide is a weak base. What is pH? Answer is pH measures the hydrogen ion concentration of the solution. The neutral value pH is 7. Less than 7 values are called acid. More than 7 values are called as base. pH equal to minus log plus H plus. What are tablet physical parameters? Answer is average weight, weight variation, friability, diameter, thickness, length, width, disintegration test and hardness test. What are the types of gas chromatography? Answer is gas liquid chromatography and gas solid chromatography. Explain about components of the gas chromatography. Answer is gas inlet, flow controller, sample injector, column, detector and data recording system. What is normal phase HPLC and reverse phase HPLC? Answer is in the normal phase HPLC, mobile phase is non-polar and stationary phase is polar. In the reverse phase HPLC, mobile phase is polar and stationary phase is non-polar. What are polar compounds and non-polar compounds? Answer is the compounds which are soluble in water is called polar. The compounds which are not soluble in water is called non-polar. In the normal phase HPLC, which solvents are using as a mobile phase? Answer is N-hexane, heptane, chloroform, methylene chloride are using as a mobile phase in the normal phase HPLC. In the reverse phase HPLC, which solvents are using as a mobile phase? Answer is water, acetonitrile, methanol, ammonia and phosphate buffer are using as a mobile phase in the reverse phase HPLC. What is the formula for RSD? That means relative standard deviation. Answer is standard deviation divided by mean into 100. We are putting means we are getting the result. What is the formula for AV value, acceptance value formula? Answer is m minus x bar plus k, that means constant into standard deviation. For 30 units constant value is 2, for 10 units constant value is 2.4. What is ICH? Answer is International Council for Harmonization of Technical Requirement for Pharmaceuticals for Human Use. How many groups are there in the ICH and what are all it is? Answer is there are 4 groups are there in the ICH. Those are quality, safety, efficacy and multidisciplinary. What is LIR and reasons for the HPLC related LIRs? Answer is LIR means laboratory investigation or incident report. LIR happens in the HPLC by leak detection, RT shifting, peak shape failing, system suitability failure and communication error. What is OOT and explain it briefly? Answer is OOT means out of trend. It is happened for the stability study products. The results are within the specification limits, but it is not meeting to the previous results. At the time we are taking OOT. These are all the top 50 interview question and answers for the quality control department. If you learn all these, definitely you will get a job in a good company. And if you didn't subscribe my Pharma QC YouTube channel, please subscribe, share and like it. If you have any doubts, please put a comment in comment section. I must reply it. Thanks for watching my Pharma QC YouTube channel.